Well, hey, all you cool cats and kittens. In this video, we're going to talk about how to clone an expression plasmid. Our overall theme here is biotechnology, and in the next couple of videos, we're going to talk about the use of biotechnology for biomedical research. One thing to point out, though, is that biotechnology has really become a part of everyday research for anything that's involving cell biology or molecular biology. This also includes microbiological and virological research. Biotechnology is the exploitation of biological processes for industrial or medical purposes. Primarily, what happens is that the processes of replication, transcription, and translation are going to be exploited. And I'm showing you here a couple of different examples by which these processes can be exploited. For example, on the right here, you're looking at two ways in which the biomedical field exploit biotechnology. On the top right-hand side, you see Gardasil, which is a subunit vaccine that is used to prevent infection of HPV. On the bottom right, you see a vial of insulin. This insulin protein would have come from a human gene, but it's being produced in bacterial cells. Before we were able to express insulin genes in bacterial cells, we used to have to isolate insulin from pig pancreas. More recently, what we're finding is that we can use biotechnology to sequence genomes and to potentially edit genomes through processes such as CRISPR-Cas. Other mechanisms employ bacteria as a way to serve as what we would call biosensors. In this example here, bacteria would sense some sort of environmental stimulus, would compute it, and then lead to some sort of response. We can engineer bacteria to respond with color changes, for example, as a way to detect toxins or other pollutants that we might have in water. So what we'll start with here is just a broad overview and then talk about the specifics. If we wanted to create a cloning plasmid or an expression plasmid containing the insulin gene, we'd probably want to work with cells from the pancreas that produce the insulin mRNA. From these cells, we would isolate that RNA and then perform a process called reverse transcription to take that mRNA and turn it into complementary DNA or cDNA. That cDNA then can be amplified to produce many copies through a process known as PCR. That cDNA and our plasmid we can cut or digest using restriction enzymes. This is basically going to create almost like puzzle pieces that would eventually be able to be pieced together. And in that piecing of the gene or our cDNA and the plasmid, we can generate recombinant DNA, creating this expression plasmid. So let's imagine a scenario in which we've already taken our cells and we've isolated from them RNA. And then we've enriched this pool of RNA to contain only mRNA. In order to reverse transcribe this mRNA into DNA, or complementary DNA, we have to use what's known as an oligo-DT primer, or a poly-DT primer. This primer is going to contain thymines, which will bind to the poly-A tail of an mRNA. The other component of this process that we're going to need are nucleotides and the enzyme reverse transcriptase. Reverse transcriptase is an enzyme that reads an RNA template and produces DNA. And it, like all other polymerases that we've talked about, is going to proceed in a 5' to 3' direction of the strand that it is making. So this particular reverse transcriptase will begin where that oligo-DT primer is located, and it will proceed toward the 5' end of the mRNA, and all the while, as it's doing that, it's going to be making cDNA, or complementary DNA. Let's take a look at what this process looks like in a bit more detail. Let's imagine here that we're going to focus in on a short segment of RNA. It reads 5' ACG UU AG 3'. Well, this reverse transcriptase is going to make the complementary strand of DNA. And so in that case, from left to right, that DNA is going to read 3', TGC, AA, TC, 5'. You'll notice that the mRNA and the complementary DNA are anti-parallel and also complementary to one another, as the name of that DNA suggests. You may be asking, where do we get this reverse transcriptase from? Well, in part, we've gotten it from biotechnology and from bioengineering. But one of the sources of reverse transcriptase is what's known as the Maloney-Murine leukemia virus, which is a retrovirus like HIV. All reverse transcriptase enzymes are RNA-dependent DNA polymerases. The next step of this process then is to make many copies of one particular cDNA. So we have our pool of cDNAs here from mRNA that has been reverse transcribed, and the first thing that we do is we go ahead and we heat the sample at 95 degrees Celsius. This helps to denature or separate those two strands of DNA by breaking the hydrogen bonds that hold together the base pairs. You can kind of think of this step as doing the same thing that helicase would do during replication in a host cell. Helicase is there to separate the hydrogen bonds that hold base pairs together, and that's what the heat is doing. And so we have no need for helicase enzyme in this PCR. In the next step, we cool down the sample to about 50 to 60 degrees Celsius, and this allows for primers that are specific to the cDNA that we want to amplify to bind to both strands of that cDNA. So if our goal here is to amplify insulin, we would design these primers so that they are binding to segments of the mRNA that is going to encode for the insulin protein. Next, the temperature of the sample is brought up to 72 degrees Celsius, and this allows for polymerization of that cDNA. And we do this process in what's known as a thermocycler, a large instrument that takes the DNA sample through multiple rounds of this 95 degrees Celsius, 50 degrees Celsius, and 72 degrees Celsius heating as a way to make multiple copies of that DNA. It's important to note some of the reagents that we need for this, though. Not only do we need a template of DNA, but we're also providing primers, which replace the need for the enzyme primase, and we're also adding our own DNA polymerase and deoxynucleotide triphosphates as a substrate for this polymerization. The first DNA polymerase to be used in PCR reactions was known as Taq polymerase. This particular polymerase comes from the bacterial species Thermophilus aquaticus, and it was isolated originally from Yellowstone National Park. Since that time, we've used different formulations of either Taq polymerase that we've modified by genetic engineering, or we've used other heat-stable DNA polymerases. Note at the temperature at which these processes are occurring. This polymerization step here is occurring at 72 degrees Celsius, and so we need to use polymerases that are going to be able to function at that high temperature. A human polymerase just wouldn't work. This image here then is showing you a broad overview of this process of PCR. You can see that in the far left-hand side, they're showing you those first three steps that we talked about. The denaturation, which is heating the sample to 95 degrees Celsius to separate those two strands of DNA. The annealing, which allows the primer to bind to the template DNA, and then the extension or the polymerization step. After 2 cycles of this, if we started with 1 single copy of our DNA, we'd have 4 copies at the end. And after 3 cycles, we'd have 8 copies. In other words, the number of copies of DNA that we theoretically would have would equal 2 to the n, where n equals the number of cycles. So if we began with one single copy of the DNA in the first round, after 30 cycles, we would have on the order of 1 trillion copies of this single fragment of DNA. Now, cloning plasmids or expression plasmids are used to express genes in bacterial cells at high levels. And so this is kind of an example of a traditional expression plasmid. Over on the far left-hand side, we have an origin of replication that allows for this particular plasmid to be replicated. On the bottom in orange, we have a selectable marker. This particular one encodes the beta-lactamase gene, which would give bacteria that contain this plasmid resistance to the antibiotic ampicillin. This allows us to select for bacteria that contain this plasmid. On the top, you can see we have a promoter where an RNA polymerase would bind, as well as a transcriptional start site. And just downstream of that transcriptional start site, we have what we call a polylinker or a multiple cloning site. This is where we would want to put our gene of interest. And so you can note here we have a number of different terms like EcoR1, EcoR5, Hindi3, and these are all examples of what we call restriction enzymes. So you may be asking, what are restriction enzymes? Well, restriction enzymes were discovered in the 1960s and were characterized through the 1960s and 1970s. When first discovered, they were found in bacterial cells, and we knew that they had a role in protecting bacteria from infection by bacteriophages. But now we use these enzymes all the time in biotechnology to generate recombinant DNA. Let's take a look at this particular restriction enzyme site for the restriction enzyme EcoR1. You'll note this sequence in the 5' to 3' direction reads GAA-TTC. And this is the case for both the top and bottom strand of the DNA. And so we say that this sequence is palindromic. Restriction enzymes recognize palindromic sequences such as this one. What EcoR1 would do is it would make a cut or a nick in the sugar phosphate backbone between this guanine and adenine nucleotide on both of these two strands. Doing so is going to linearize this DNA, and they're both going to contain overhangs that begin with AATT. These overhangs are sometimes called sticky ends. Other restriction enzymes will create what we call blunt ends, where there is no overhang like the ones shown here. So let's take a look then at how we can take these restriction enzymes and use them to help us create this recombinant DNA. We are going to use the restriction enzymes EcoR1 and Hindi3, which recognize two different palindromic sites. You'll see that sites for these restriction enzymes are found not only within our multiple cloning site of the expression plasmid, but they are also found within the PCR product of our cDNA. On the bottom of the PCR product here, you'll notice that I've labeled the sequence that would code for the start and the stop codons of the mRNA to be produced by the cDNA. Cutting the plasmid and our cDNA with the restriction enzymes EcoR1 and Hindi3 are going to create sticky ends on both of these DNA fragments. This allows us then to orient the cDNA into the plasmid in the correct direction or orientation so that we'll be able to produce mRNA from this cDNA. Now, it's important to note here that the EcoR1 sites of the cDNA and the plasmid are sticky ends, and they are complementary to each other. That same thing is true for the Hindi3 sites of the plasmid and the cDNA. And so this is going to allow for these two fragments to stick or base pair together. However, it does not create the covalent bonds in the sugar phosphate backbone that are required to permanently hold together the cDNA and plasmid. To do that, we need to use the enzyme ligase, which, as you will recall from our conversation on DNA replication, is used to glue together or to paste together the Okazaki fragments by creating covalent bonds in sugar phosphate backbones. we would use that same enzyme here to create a recombinant plasmid now containing our gene of interest. So let's go ahead and review this process. We talked about how mRNAs can be reverse transcribed using an oligo-DT primer and reverse transcriptase. The particular primer that is used here allows us to reverse transcribe all mRNAs into cDNA, or complementary DNA. And that cDNA then, we can go ahead and amplify one specific cDNA using a process that's known as PCR, or polymerase chain reaction. The amplified product from PCR can be digested with restriction enzymes, and we can also digest our cloning plasmid with the same restriction enzymes. That allows us to recombine these two fragments and then to essentially create a closed-loop plasmid containing our gene of interest by using the enzyme ligase. Now you may be asking, how do we know if we've got the right sequence, and how do we actually express this plasmid? Well, that's for a later talk. In the meantime, take care.